In this video we will discuss about a new paper that discusses about this antigen specific T cell response. So this T cell response can be specific and non-specific, and in which conditions this T cell response is more enhanced. So from their previously published protocol, what it is, the idea is that they express the antigens on the tumor cells, and then they also use the CAR against those antigens in the T cells, and then check whether this T cells is able to detect those modified tumor cells and then kill those tumor cells or not. So that protocol they have already developed in their previous paper, and in this paper their starting point is to use that protocol to make melanoma cells that can express HLA-A and this MART-1 antigen, and at the same time they transduce the T cells against these MART-1 receptors, and then co-culture them for 4 hours, and thereafter they find out that there are some populations that is both tumor cells positive and also the T cells positive. And thereafter, when they did the microscopic analysis, they are able to see that these clusters are the formations of these T cells and the tumor cells. It can be seen here. And this phenomena, they checked it for the other cancers, not just for the melanoma cancers, and they are able to see that the cluster formations are seen in all the in other 4 cell lines that they have tested, and even in the other cancer cases that is shown in this panel C. Thereafter, what they did is they wanted to check, or they wanted to do the studies in a more biologically mimic conditions. So what they did is they used the different ratios of these T cells, that is some T cells that are specific to this MART-1 antigens, and some T cells are not specific. So the initial case is that they use 60% of the non-specific, 60% of the non-specific T cells and 40% of the specific T cells, and then co-culture them with the tumors. And what they're able to see, they're able to see that a 2.7-fold increase in the specific cluster formations. Thereafter, they did these studies at different ratios of the specific and non-specific, and even at like 1% of these specific T cells are able to enhance the cluster formations of the specific T cells and the tumor cells. It can be seen in this panel F and the last row, where they use 1% of the specific T cells and 99% of the non-specific T cells. And when they did their studies in the reverse order, that is, they this time keep the specific T cells higher and non-specific T cells lower. In that case also they see the similar things, that the the cluster formations of these specific T cells and the tumor cells increased in almost all the cases where they used. So in this case they used 95% of the specific cells and 5% of the non-specific, and they use it in the different cancer cells. And in all the cases they are able to see the increase in the cluster formations for the specific specific T cells compared to the non-specific T cells. So that can be seen in this panel G, where they use 95% of the this non-specific, where they use this 95% of this specific T cells and 5% of the non-specific T cells. And thereafter, once they find out that, or once they are convinced that this phenomena is true in most of the cancer cases, they wanted to do the in vivo studies in the NSG mice model, where they did the studies in like 2 different conditions. First they use the singlet T cells, mixture of singlet T cells, and second one they use the clustered specific T cells. And what they are able to see in the results that is shown in this panel I is that the case in which they use the cluster T cells are able to suppress the tumor volume, whereas in all other cases the tumor volume is not suppressed and is close to the control, but they don't use any T cells. that is they used T cells from the singlet, and also all the T cells. Even in these three cases, in these two cases, results are similar to the control. Whereas in the case where they use cluster T cells, the results shows the tumor volumes are able to suppressed. This indicates that. So this indicates that the the T cells responsive can be higher, or the cluster T cells with the tumors has a more immunogenic, can trigger more immunogenic response compared to the singlet T cells, as they need time to interact with the tumors. Whereas cluster T cells are already immunogenically active compared to the singlets. This is what they finally conclude from these results, or the results that are presented in this results that are presented in this paper or in this figure. That's all for this video. We will discuss other results that are presented in the main figure of this paper in our subsequent videos. So that's all for this video. Thank you for your time.